Hi, I'm James Bevington, a researcher in Mike Manefield's lab and commander of Mission 5 for High Seas. High Seas is an 8-month Martian analog study with a crew of 6. We maintain a 20-minute communication delay, which is 40 minutes round trip, and we live in a small habitat on the slopes of Mauna Loa volcano in Hawaii. Now I'm going to suit up for an EVA and show you around outside. Every time we go outside, we have to wear an EVA suit. So this includes GoPros for the researchers, radios so that we can communicate with Habcom who stays back in the habitat, battery packs to power our fans for ventilation, and water for hydration. Then we seal up the suit so that it's airtight. Before we go out, there's a short decompression to equalize the pressure between the hab and the Martian environment. In the distance you can see Mauna Kea volcano, which is right across from us, and there's our storage container where we keep excess items that we don't want in the hab. So our primary source of power here at High Seas is a solar panel and a battery system, but we have this propane tank that we use for hot water and for heating. We also use this propane for our backup generator system. So for our mission we've used about 30% of this tank, but we have a lot extra just in case as backup. So now I'm going to take you to our secondary backup system. Now we'll climb back up the side of Mauna Loa volcano, which at 2,000 meters can be quite breathtaking. So this is our secondary system. It's a hydrogen fuel cell. Now this system isn't strong enough to supply our primary use, but it's strong enough to supply our baseline in case the primary backup goes down. This will keep the hab up and running until we can get the primary generator going again. I hope you enjoyed the tour. As you can see, we use a lot of resources that could easily be substituted with methane. The generator and the fuel cells could both be replaced with methane utilizing counterparts. Also, the required mass is quite substantial, so anything that can be produced on surface would provide a great benefit, and that's exactly what we're trying to do in Mike's lab. MMARS stands for Microbial Methane Associated Research Strasbourg. This is an experiment that started as my personal project when I was a master's student at the International Space University, and has since grown into a multi-flight campaign. The science team consists of the International Space University, the University of Strasbourg, and more recently UNSW. Hardware is being provided by Airbus Defence and Space in Friedrichshafen, Germany, and we have 2 launch partners, Space Tango, a small company in the US who aims to send small payloads to the International Space Station, and Ice Cubes from Space Application Services in Europe. Primary funding source is the city of Strasbourg, and our goal is to investigate microbial methane production in space, i.e. biogas in space. Our current model for organics on Mars can be summarized in the top left corner of this figure. The idea is that complex organics come in from cosmic dust and meteorites to the surface of Mars. These complex organics are then broken down via ultraviolet radiation into smaller pieces. Starting with the composition of the Murchison meteorite, Benner et al. 2000 took what we know about the Mars environment and photochemistry and made predictions about how the complex organics in the left column would break down on Mars. And as you can see, they predicted that most things would break down into either a benzene ring or acetate. So recently, Curiosity rover confirmed the presence of benzene rings. Acetate has yet to be found, but we believe that we're on the right track at the very least. So this is a very useful column for us because on the left side we have what we'd expect to find on an asteroid in space, and on the right side we have what we'd expect to find on the surface of Mars. On Earth, complex organics can be broken down by anaerobic digestion. So the basic process is that you start with something complicated like chitin, and you break that down into simpler parts that eventually end up as acetate. From there, the final step is to convert acetate into methane. So the first three steps here can be accomplished by one organism, but the last one has to be accomplished by a second organism known as a methanogen. MMARS-1 started because Airbus was interested in learning how to do things in the new space era. So the old school way of doing things was to define an experiment, then build very customized hardware and spend several years testing that hardware for your one experiment. But with new companies who are sending small payloads, there's a different opportunity, and the different opportunity is to build a simpler payload but do much faster iterations. So rather than doing all the testing on the ground, you might as well flight test it. And so that's kind of the new idea. So Airbus formed a partnership with International Space University trying to test a turnaround in 6 months, and that's even fast for the new space world. So the primary goal of the first one was to do a hardware shakedown. So we had to design a new sensor system to be able to test methane production, and Airbus also built a hardware interface that allowed them to take some old payloads that they had from the shuttle days and be able to use them in a 1U payload. So this flew in February of this year. We included just methanogens, so the last step of biogas formation, and it's because the sensor system only detects that last step. We also included a lot of controls to help us determine exactly what was going on. MMARS-2 is currently in preparation with an expected launch date in February on board a commercial Resupply 14 mission from SpaceX. We will investigate the independent methanogenesis step, which is relevant for the surface of Mars, and also the entire anaerobic digestion process, or the coupled process, which is relevant for an asteroid or a waste stream, either in a deep space habitat or on the surface of Mars. The expected outcomes are to validate the concept of biomining in microgravity, and second, to inform the in-situ resource utilization community on ideal production. So this experiment will be on board ISS and will have good temperature, lots of media, and things that microbes really like. And so this is sort of the upper limit of what you might expect to be able to produce in space. And with that, thank you for your time, and so long from the high seas habitat.